Come on, man. There you go. Let me get this out the way. Is it time to start? I think it's time to start. Let's see. I'm still admitting a couple of people here. Not very many. There's only 10 of y'all so far. And it is time to get started. There's some more people. People are just being late today. Okay. Let's see. There's more. Tardy. You guys are all tardy. Am I on there? Yeah, I'm there. Okay. You guys hear me okay? Today is Tuesday, January 19th, 2021. It is now, about a minute or so after 1 o'clock. So we're going to go ahead and get started. But hey, I want to show y'all something. It's our first lab, I guess. Rural lab. Let me show you this. This is your course policy sheet. Remember I went over this the first day, like in a PowerPoint. You can print out this if you need to. It's on my courses under the syllabus and course policy link. Anyway, this is a nice, convenient, handy dandy thing to have to know what we're doing every week. So this is week 2. We're doing cells and tissues, the cells and tissues chapter, and concentrating on animal tissues. There's all kinds of other things in this chapter in your atlas, but that's what I want to concentrate on here. And next week we're doing perpetuation of life and so on and so forth. Okay, so that's where we are. And so today, what I've done is I turn this lab, and that's pretty much how I've done all the labs, is I've turned them into to PowerPoints to present the material. Most of the labs anyway, I've done that. Let's see. Wait. Okay, that's what you guys are seeing. I suppose. Okay, I can do that. All right. Oof. Let me just tell you, give you a little background on this lab and how I used to do it. Is remember, I'm Zooming the first hour of lab, and then that's it, an hour-ish, I think. I'm gonna try to keep it about an hour under, so I might go a little bit over. Maybe sometimes I might go a little under. I guess that's okay too. I don't think anybody complain about it. But I wanted to let y'all know that remember the last 2 hours of lab, if for some reason you wanted to come to lab face to face, you could, but I'm not going to do anything different than I'm going to do. So I'm not going to show you anything different. So what are we going to do? I don't know, you know, maybe if you have questions about something, or you just want to look at something, whatever, let me know. But I'm going to ask you at the end of the Zoom, you know, who wants to go to the lab, and I'll make sure that I show up, because if I don't have any students going to show up to the lab, I'm not going to go and sit in there for 2 hours by myself. So anyway, let me know, and we'll talk about that. So what do we do? So this is really not going to take 3 hours. You know, like in the regular semester, you know, students would be working with microscopes and fumbling through slides and all kinds of stuff. And so I've taken all that out of the equation. So it's reduced down quite a bit. So you know, we can basically zip through this animal tissues with this PowerPoint I have. And so what I would do traditionally is, to study animal tissues, I would take sections from an animal, and our subject of choice, our study specimen was a mouse. So I take pieces of the mouse and look at the structures or organs, and then look at the tissues that are in there. And this, what the students would do is, where there were 3 slides, and we would look at the slides and of course identify the slides from different parts of the animal, different parts of the body, and then look inside and try to figure out what kind of tissues we're looking at. Okay, and so, like I said, I've taken a lot of that work out of the lab, and took pictures of everything and show you. So there's 3 slides. We're going to start with our first slide right here. And what you have here, let me get my laser pointer here somewhere. This is a cross section through the mouse's tail. Okay, and you know, the tail is I don't know if you think of it as an organ. It is an organ technically, because what an organ is a structure that's made out of different types of tissues. And you can see this is underneath the microscope, magnified about, this is about 40 times, I think. And you can see all the different structures inside the tail, and all the different structures, the colors and the patterns and the textures actually equate to a different type of tissue. And so if you kind of just think about it, you can probably just guess, you know, and figure out, you know, what the tissues probably are. You know, the superficial most tissue, you know, the outside of a mouse's tail, that's where the skin is going to be, right? And then the very, 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 very center, what's in the middle of the tail, you know, it's bone. And so actually what we have here is the caudal vertebra, you know, the little bone that's in the tail. But this is a young mouse, a very immature mouse, little pinky mouse. You ever heard of little pinky mice that some people use to feed their pet snakes and so forth? Anyway, this is a newborn mouse. And so, and like in most mammals, when the skeleton is immature, it's not fully bone yet. It's cartilage before it's bone. And so we're going to see that this vertebra in here is made out of a connective tissue, not bone, but cartilage. But it's eventually going to become bone when the mouse, you know, matures. Anyway, so skin on the outside, skeletal structure on the inside, and all kinds of other tissues between the two. And what would be between bone and skin? Well, muscles and tendons and so forth. So we're going to look at all that. So that's what we do. We're going to proceed, first of all, identify this as a tail. It's a mouse's tail. And then look at the tissues in it. So I think what I'm doing here is I'm pointing out different structures and identifying the different types of tissues, okay? So the first thing I'm pointing out, you can kind of see on the outside, it's kind of shaggy looking, dark stuff going around. That's the epidermis. The epidermis is the superficial most layer of your skin. So skin, epidermis, same thing. And so the epidermis isn't a tissue, isn't the name of the tissue, it's a structure. And, you know, different structures made out of different kinds of tissue. So what's the epidermis made out of? So you zoom in on it, magnify, you go from like 40 to about, this is probably about 400 times. This is zoomed in of the epidermis, and you can see that this is in fact a specific type of tissue. this tissue right here, you can see that these are individual little nuclei of cells. They're going up, 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 up like this until, and then you kind of get this layer right here where they become real thin and flat and dead. This dead layer of cells right there. This is called stratified squamous epithelium. Okay, that's the name of the tissue. So the epidermis of the mouse's tail is made out of stratified squamous epithelium. And then look here, in parentheses, I have the word keratinized. You see that? The word keratinized just means, if something is keratinized, it means that it possesses the protein keratin. So, well, keratin is a very tough protein. It's the dry protein that makes up the structure of your fingernails and your toenails and your hair. And it's also, it makes sense too that it makes the dry membrane on the outside of your skin. And you can identify this stratified squamous epithelium as being keratinized because you can see that it has this kind of detaching dryish membrane. Well, that's due to the protein keratin. So anytime you see it like this, dry and detached like that, it's a keratinized epithelial tissue. And this specific one, it's called stratified squamous epithelium. Okay, the word stratified means that it's in layers, and anytime you got two or more cell layers thick, and you can see that these are several cell layers deep, you know, it's stratified. And the word squamous means just flat. So these cells, when they mature, obviously when they're dead, they're very mature because these are old cells. They're flat. So several cell layers of flat cells forming an outer membrane, an epithelium. Epithelial tissue is a class of tissue, and it's what lines and covers the outside of structures. And so here, covering the outside of the tail, it's epithelial tissue called stratified squamous epithelium, and it happens to be keratinized. See that? Hey, that's our first tissue. How about that? Okay. What's next? Well, I'm backing away. So you zoomed in, now you can zoom back out. And I want you to see where you were. See where you're out there. See this kind of shaggy layer? That was the epidermis. Now underneath the epidermis is the dermis. That makes sense, right? The epidermis and dermis, you know, those are layers of the skin. The word dermis refers to, well, not the epidermis, which epidermis means upon the dermis. So under the dermis, that's the next tissue layer that's deeper. And you can tell that this kind of, all this reddish area, see where I got my arrow going all the way around? It's kind of clear, it's not so shaggy. You know, we're gonna zoom in on it and you'll see it a little bit better. But that's the dermis of the skin. That's the same layer of the skin that leather is derived. Like, you know, leather from a hide, you know, you got like hair and superficial skin cells on the outside. Then you got leather, the hide, and then underneath the hide, you got the fat layer and so forth. Well, the dermis, you know, that's like the leather-like area. So we're going to zoom in on that and see what kind of tissue it is. And so it's going to be something different. You can kind of tell that it looks different already. And so what do we have here? Well, remember, this is where we were. Now we're underneath. This is the dermis. And you can see it's not epithelial tissue. Remember, epithelial tissue is always the stuff that's on top. It's always going to be exposed to the free surface here, you know. So this is underneath it. Underneath epithelial tissue, you're always going to have a connective tissue. And this connective tissue is a special kind of connective tissue that's just jam-packed, full of irregularly arranged collagen fibers. Collagen, it's like the most prominent type of connective tissue or connective tissue fiber in your body. It's everywhere. It's very tough, reinforces structures. Because you think about leather, you know, it's very fibrous, right? There's a fibrous connective tissue called dense irregular connective tissue. Dense because it's jam-packed full. You see all this reddish, kind of looks like shredded wheat in there. See the red fibers, and then you see these dark purple dots everywhere. Those are the cells that form and maintain those collagen fibers. These are called fibroblasts. So you got a collection of fibroblasts with densely packed collagen fibers everywhere, but they're irregularly arranged. So dense, irregular connective tissue. And so that's what you got. This is your second tissue. So remember you had stratified squamous epithelium, there's keratinized here. Now underneath here, forming the dermis of the skin, dense, irregular connective tissue. Okay, you see that? All right. Second tissue down. Now look. Going a little bit deeper. See, remember our arrow was here, now it was here, now it's here, the third layer right here. See the white area they're showing you? All right, just ignore this white block right here. I just kind of deleted something. I hijacked this slide from somewhere. It was saying something here I didn't want you to see, so I just deleted it. But anyway, but it's a great slide. I really like this slide. This kind of whitish zone, kind of just like space. This is another region, you know, the mouse's tail, you know, you got epidermis, dermis, and then underneath the dermis. Sometimes they call it hypodermis, but here I call it the subcutaneous layer, which literally means under the skin. And by the way, that's what hypodermis means too. Hypodermis means under the dermis, or subcutaneum. Your cutaneum is your skin, so subcutaneous means under the skin. So there's this layer underneath the skin. And so what's under your skin? Fat, usually. You know, you get subcutaneous fat. And so fat is a connective tissue. It's not like a fibrous connective tissue with collagen fibers and real, you know, strong and durable. Fat's really not like that. Fat is more cushy, right? It's soft. And so it has other properties, and your body uses it for different things. And so it's going to look different. And so it looks, under the microscope, it always turns out being really thin and delicate, because the fat cells are delicate, but they contain, what their job is to do is to sustain a fat droplet, a lipid droplet. And your body uses that lipid for different things. And so you see, this is typical of mammals. Mammals have a subcutaneous fat layer, and then in that fat layer, the blubber layer, if you want to call it like that, is there to help insulate your body against heat loss. And so yes, you know, you want to do that because everybody knows that mammals are warm-blooded, right? You got warm-blooded animals, cold-blooded animals. Well, if you can be warm-blooded, which means, or we call it homeothermic. Homeothermic just means you have a constant, unchanging internal body temperature. And so if you have your internal temperature is going to be, you know, kind of stabilized, like your body temperature is 98.6 degrees, right? If you don't have fever, right? Or 37 degrees centigrade. And it typically stays there, and you have mechanisms in your body to make sure that it, you know, doesn't get too high, doesn't get too low. It stays right around 98.6. Well, if you have anything that you wanted to keep a sustained temperature, or homeothermic, that's another way of saying it, you want to insulate it. Now, fat cells and the lipid, it's used for all kinds of things. It's used for stored fuel. It can cushion things, and you can do all kinds of things. But just the nature of it, it helps insulate against heat loss. So it's a good thermal insulator. So mammals, you know, all kinds of vertebrates have fat, right, or fat cells. But mammals in particular develop subcutaneous fat to aid against heat loss. Okay, and that helps, you know, keep them warm body temperature. Anyway, this is what it looks like under the microscope. That's what you're looking at. This very thin, delicate layer. So what I'm going to do, obviously, is I'm going to zoom in, and we're going to look at some of these fat cells under the microscope. And look, there they are. Well, here's a couple little patches of them right here. You see some here too. Real thin, delicate area, and they look like little bitty, like dried soap bubbles, or look like you're looking through some chicken wire or something like that. Now, there's all kinds of other random stuff out here. in the field of view. Just ignore that for right now. We'll get to them. But here I'm just going to show you this, our next tissue, which is going to be the fat, or it's a type of connective tissue, and fat is known as adipose connective tissue. So you get a little patch of adipose right here, a little patch of it right there. And I think my next slide is going to get a better view. Well, maybe not. I thought I was going to show you something that looks a little bit better. But we'll see it again. We'll see fat in some other slides too. Let me page up for a second. Okay. So that's adipose connective tissue, usually in the subcutaneous area. There's hypodermis, right? Okay. Now, what was our third one? Now, here's where we were. Now we're going right there. You see all these red ball-looking structures here, there. There's some right here, some right there, and some right there. What is all? Those are all tendons. Okay, you know what tendons are, right? Tendons are what muscles use to connect to bone. Okay, you know, how muscles work, you know, they contract. And of course, when they contract, they pull, and they pull on some kind of substrate, usually the skeleton, right? And so the muscle rarely just connects directly to a substrate. It usually uses a connective tissue to anchor to whatever. And so when it uses bands of collagen fibers, it's collagen again, to anchor to something, it forms a tendon. And so the muscle pulls on the tendon, and then the tendon pulls on the skeleton. And there you have it. This is what it looks like in cross section. And so these are cross sectional views of tendons that are coming down and connecting to the tail. You know, these are caudal tendons or tail tendons. And here they are zoomed in this is probably about 400 times. You can see these are tendons in cross section. And this is it's all collagen. It's another connective tissue, but this time it's called dense regular connective tissue. Remember we had dense irregular in the dermis of the skin? Here's dense regular connective tissue forming the tendons. And so why do we call it regular opposed to irregular? Well, remember, irregular was the irregularly arranged collagen fibers, just kind of going every which way. Well, here, all the collagen fibers are running the same direction, and then they're cut in cross section, so it just looks like these little bands. You see that? They're not going this way, they're not going that way, this way. They're all coming straight at you. So they're cutting into, it's like little rods. You're looking into the ends of them. And so this is what it looks like. These are the cross-sectional view of tendon fibers. And the little black dots you see in the center of them scattered everywhere, those, again, are the cells that are associated with the collagen fibers, fibroblasts. Okay, so you have fibroblasts in dense regular connective tissue, and you have fibroblasts in dense irregular connective tissue. Essentially, anywhere you're going to have collagen fibers, you're going to have fibroblasts. Okay. Well, this is what it looks like forming the tendon. And I'm just going to let you in on some inside information. You're never going to have dense regular connective tissue anywhere else other than forming tendons or ligaments. They form tendons and ligaments, and it looks pretty much the same every time, like a cross-sectional view like this. Okay? So there you go. Some people coming in still. Welcome. You're really tardy. You already missed almost everything. All right. Oh, and it's just another view. I'm just showing you some more dense regular connective tissues, a couple more tendons right there, just from a different view. Okay. We've seen this view before. Okay. Now, that was tissue number 1, 2, 3, 4, right? This is our 5th tissue right here. You can tell that, see this patch right here? There's another patch right there. Here's a patch, there's a patch, there's a patch, and so forth. We had the structures. What are the structures? Your epidermis, dermis, subcutaneous tissue, tendons. Now, what's this? Well, this is muscle. That's the tail muscle, or just muscle. What's muscle made of? Well, you have 3 different types of muscle. Okay, if you look at animal tissues, there's cardiac muscle in the heart. There's smooth muscle in hollow organs like your stomach and intestines and so forth. And there's skeletal muscle associated with the skeleton. And sure enough, you're getting close to the skeleton here. Here's the vertebra, that's the skeletal element here, and this is the muscle associated with it. And so that's going to be skeletal muscle. Now, all three different types of muscles, you think about them, they all have different functions. Skeletal muscle is to operate on the skeleton, allow for movement, right, of the body. Smooth muscle is to, so the organs can move, like in your stomach, it helps the food churn in your stomach, and in your intestines, moves the food by peristalsis, and in your bladder to help empty your bladder, or in your uterus to help push a baby out, or whatever, all your hollow organs have smooth muscle in their walls. Different function. Here, skeletal muscle to move. The cardiac muscle, of course, is to move, blood to, so the heart can contract. So all three different types of muscles have three different functions, and of course they have three different morphology. They all look different, and you can identify that underneath the microscope. So plus you're looking at this. This is not a hollow organ. It's a solid, organ. It's got bone in the center of it, or skeletal element, I'll call it, because it technically isn't bone yet. So it's not a hollow organ. It's not the heart. You would expect skeletal muscle, and if you zoom in on it, it's going to look different than the other. Well, we're going to look at the other types of muscle too, and you'll see this difference. So how do you know this is skeletal muscle? Well, let's zoom in on it, and here we are. You're looking at it. Here's the tendons, and it's usually adjacent to the tendons because it makes sense. The muscle's going to pull on the tendon's going to pull on the skeletal element. And it looks kind of like dense, regular connective tissue, but it's not. You look at the detail, it's a little bit different. You've got these little bitty, kind of triangular, some of them are square-like, but very rigid angles, sharp little structures here. Each one of these little red little squares or triangles, whatever shape they are, polygons, each one is a single skeletal muscle cell. So all of this is skeletal muscle, and these are individual little cells. And this is only magnified probably about 200 times. You zoom in on it more, you'd be able to see individual nuclei. I can't remember if I zoomed on this more or not, but we'll see. But this is all skeletal muscle. Just don't confuse it with the tendon, which is dense regular connective tissue. Let me see. Do I zoom in on it? No, it's a different view on it. This is a good view. I'm showing you this because, see, these are individual little skeletal muscle cells. And you can see the tiny little black dots on the edges here, there. See, each one of those little tiny little black dots is a nucleus. So if you think about it, here's one cell right there, and there's a black dot there, black dot there. See that? There's two of them. Maybe I should have a pointer. Do I have a pointer? Laser pointer. I turned it off. Need something with some more. Unless I just turn it off and use my. Yeah, see, you got my arrow, my cursor. Make it a little more pointy. At the very tip right there, little black dot, little black dot. Each one of those is a nucleus. So this cell has two nuclei. This cell has a nucleus. This cell has two nuclei. This one has two. This one has one. Well, this one has three. Look right there. 1, 2, 3. The point I'm trying to make is that each one of these is a cell, and there's more than one nucleus per cell. Then these cells are multinucleated. You hear me? Multinucleated. What does that mean? Well, it means the cell has more than one nucleus. Okay? And only skeletal muscle is multinucleated. The smooth muscle and cardiac muscle are not going to be multinucleated. And you can tell. So if it's multinucleated like this, okay, that's skeletal muscle. It's a good hint for you. So I'm trying to share that with you. Okay, so skeletal muscle. Now finally we get to the big old arrows. That's for all of this. That's why I'm a big fat arrow to show you all this big structure right here is the skeletal element, the wannabe bony vertebra in the tail. Here I have just tail vertebra. Now I say wannabe bony because normally when you think of your skeleton, you think of bone, right? Well, your bone isn't bone. Doesn't start out as bone. Most of your skeleton is cartilage before it becomes bone. It kind of uses cartilage as like a little template before it turns to bone. So often, not all your bone, but a lot of it starts off as a cartilaginous template and then it ossifies or it turns to bone. It just kind of it incorporates minerals like calcium phosphate, calcium carbonate, some other minerals to harden it, okay, and make it rigid like the characteristic bone you're familiar with. Well, there's 3 different types of cartilages, okay. There's just regular standard old gristle, like you would think, cartilage, which is called hyaline cartilage. And then there's modified forms of hyaline cartilage. And it's modified in the sense that it incorporates 1 or 2 different types of collagen fibers. If hyaline cartilage is modified with elastic fibers in its matrix, we call it elastic cartilage. If it's modified with collagen fibers in its matrix, and when I say matrix, I'm talking about just in the cartilage, the extracellular space and material, okay? But if it's modified with collagen fibers, we call it fibrocartilage. And so hyaline cartilage, elastic cartilage, and fibrocartilage, okay? Hyaline cartilage, the type that's the standard unmodified form, is by far the most abundant. This is the cartilage that forms the fetal skeleton for the most part. Most of your bones is hyaline cartilage before it becomes bony, okay? And that's what you're going to see here. Now, does hyaline cartilage exist in the adult? Yeah, you still have hyaline cartilage in your body doing other things, like in your joints where one bone meets another bone, you retain hyaline cartilage there, and it cushions the ends of those adjoining bones. And then that forms the cartilage in your nose. It forms the cartilage like that's in your ribs and so on. And the other types of cartilages have other special functions based on their location, okay? So, but right here in the center of the tail, where you got the tail vertebra, the caudal vertebra, if you want to call it that, you have hyaline cartilage. And so here it is zoomed in. It's about 200 times. And you can see this tissue right here. See, it kind of comes up as a point like that. That's the tip of the vertebra right there. And you can see that it cells in it. It's just very distinct looking, okay? This is magnified, like I said, only about 200 times. We zoom in on it a little bit more. You can see some more detail of it. I think the next slide is probably gonna be zoomed in some more. It's hyaline cartilage, I hope it is. Oh yeah, this is zoomed in about, this is probably 400 or 500 times, you know, about twice as much as the last slide. And this is a beautiful section of hyaline cartilage. Remember, there's hyaline, elastic, and fibrocartilage. But this is the regular unmodified stuff, okay, that you have in the tail, right? And so I don't see any fibers or anything. It's almost kind of clearish. You see, it's like a stained glass window. As a matter of fact, the word hyaline means glass, and so it is kind of like a stained glass window. And so you see there's these little pits, these little spaces, and inside the spaces you have a cell. So this is, it's like a hole, and then there's a cell, and this is the nucleus of that cell. The cells that are in there, those are the cartilage cells. We call them chondrocytes. So it's a chondrocyte inside that space. The space itself is called a lacuna, and I think I labeled that. Watch this. Yeah, see right here? Lacuna is the space, and the chondrocyte is the cell that's inside of it. And this is the only kind of tissue that does that. Cartilage is the only type of tissue that does that, where you have chondrocytes in lacunae. It's arranged like this because you know, cartilage. It's very, very dense, you know, and firm. It's a firm extracellular material, or we call the matrix of it. Okay, so it's very, very characteristic. Okay, so there you go. Hey, you know what? That's our first slide. That's the esophagus. I mean, that's the tail. And remember the tail, I said it's like a solid organ. It's not hollow, okay? And so we looked at it. And so like you might have figured out, are we going to look at a hollow organ? Yeah, so that's why I took the mouse's esophagus. It's a hollow organ, and we're going to look at it. And so you see this, the hollow space, of course, the esophagus. This is the tube that connects your mouth to your stomach. And here it is from a little mouse. That's where his food goes, down this hollow space. And then, of course, this is the outside of the esophagus. And so, of course, you got tissue out here all the way to the inside. So the inner lining, you know, is kind of tissue, and then also the tissues, different stuff. So let's look at it. So here's looking at the inner lining. This is the space, that hollow space. That's where the food goes, which actually in microscopy, we call that the lumen, L-U-M-E-N. The lumen is the hollow part of an organ. And lining the hollow part of all organs, it doesn't matter if it's an esophagus or what, if it's hollow, it's gonna be lined with a mucous membrane, okay? And in biology, we call mucous membranes mucosae, or this is a mucosa. So mucosa, mucous membrane, same thing, but this is a special type of mucosa. The mucosa lining the inside of the esophagus, so we call it the esophageal mucosa. How about that? So it's kind of technical word for it. So here's another view, zoomed in on the esophageal mucosa, looking at it, and it's just like what you saw on the outside of the skin. Remember, you're always going to have epithelial tissue lining. This will be the top layer that's next to a free surface, and so that's what this is. This is the esophageal mucosa, and if you look, it's several cell layers thick, you know, of flat. You look kind of carefully at these ones up here, you can see they're flat. So it's just like the skin again, where it's stratified squamous epithelium, but now I have in parentheses non-keratinized. It's non-keratinized because you don't see that dry, flaky layer. Remember that flaky layer you saw on the epidermis? You don't see that, and so that's kind of like a giveaway for you that this thing isn't dry and flaky. And you wouldn't want a dry, flaky membrane inside your body. The only place you're going to have keratinized epithelial tissue is on the outside of your body, so your epidermis. So inside the body, it's always going to be non-keratinized. So here's stratified squamous epithelium, non-keratinized. So you see, so we've seen two different forms of stratified squamous epithelium, okay? One on the epidermis and then one on the esophageal mucosa. Now, I'm going back out, and we're going to go a little bit deeper. Remember, you have epidermis and dermis. Now you have mucosa and submucosa. And that's this. Here was the mucosa from here to here. So what this looks different. What's the difference between this and this? Well, these cells are dead. These cells are alive, but it's all the same tissue. But all this stuff underneath, you see that? That's all submucosa. And again, the submucosa is a lot like the dermis of the skin. Okay, same tissue, but different, forming a different structure. Okay, one's forming one of them's forming dermis, the other one's forming submucosa. Same tissue, though. It's dense, irregular connective tissue. Same stuff. Remember, it's irregularly arranged collagen fibers with fibroblasts and all that kind of stuff. So there it is right there. It's what it looks like in the esophagus. All right. Zoom back out. Look at another tissue. All this from here out. See, it's wrapping around. That's muscle. Okay. Now we've seen this already, and you would think, remember, it's kind of triangular, boxy, and the dark little dots, those are the multinucleated cells. Now you would think skeletal muscle. Why would you have skeletal? Isn't this a hollow organ? Isn't it an organ, an esophagus? You know, wouldn't that be like anything else that would have, you know, smooth muscle in the walls? Well, ordinarily you would. But the esophagus is weird, and that's why I'm looking at the esophagus, because it's kind of an exception. The esophagus has three different zones, okay? It has a zone that's connected to the mouth. It has a zone that's connected to the stomach, and it has a zone in the middle, okay? the zone that's connected to the stomach is all smooth muscle in the wall. The zone that's connected to the mouth as skeletal muscle. Because the difference between smooth muscle and skeletal muscle functionally is that skeletal muscle is voluntary muscle that you have conscious control over. When you swallow, gulp, when you're swallowing, that's a voluntary action. But once after that, your smooth muscle takes over, involuntary contraction. Like you can't control how the stomach muscles churn your food or how peristalsis moves food down your intestines and so forth. That's involuntary smooth muscle. But the zone between the skeletal and smooth is like a transition. So you got a little bit of both. Well, so here, if you look at this, I don't see any smooth muscle. This is all skeletal muscle. And so this is the portion of the esophagus that's attached to the back of the oral cavity, the throat, basically. Okay. So this is all skeletal muscle. We'll see some smooth muscle elsewhere, but there's that. Zoom back out. And then right here, it's real thin, delicate stuff. Fatty capsule. A lot of your organs are packed in fat in this kind of layer of fat. And remember, you had subcutaneous fat is kind of served as, well, you can say fuel reserve. Anywhere fat is, it can act as fuel reserve. But ideally, in subcutaneous fat in mammals is for thermal insulation. Here, this is not for thermal insulation or fuel reserves, not particularly. But when you have fatty capsules like this, it's like packing peanuts. So it's a different type of insulation, not thermal insulation, but mechanical insulation to insulate against mechanical trauma, you know? So it's kind of like packing peanuts. You know, you put packing peanuts in a box to protect something that you're shipping, right? To keep it from getting jarred around. And so there you are. And then I think I zoom in on that. Yeah, right here. This is much better than what you saw on the tail. This is the fatty capsule, and you can see these are individual fat cells forming that adipose connective tissue. I think I zoom in on that, or do I? What is this? No, maybe not. These are individual adipocytes or fat cells, and just collectively, all of this is adipose connective tissue. Remember I said it kind of looks like dried soap bubbles or chicken wire. Some people say it looks like little signet rings, a whole bunch of little signet rings together. Whatever it looks like to you, you know, that's an adipose. Now, here's just another view, so you can kind of see on some things going on. Here's your epithelial tissue. Remember, it was stratified squamous epithelium, and then it's all submucosa. Here, lamina propria, the same thing as submucosa. And then jam-packed down in there, some cross-sectional view through some blood vessels, and then you can see some little glandular tissue right there. So I'm going to zoom and see what some of that is. Your capillaries, you see that? A capillary is a small blood vessel. It's going to be itself, you know, all blood vessels are like hollow organs. And so the wall of the hollow organ is going to be lined with epithelial tissue. So we're going to see what that looks like, okay? So here it is zoomed in. This is a duct of some sort, but here's a little venule, a little arteriole. These are what connect to capillaries, but they're hollow organs. This is a hollow space. That's where the blood goes, and here's where the blood goes. Well, that lining around the inside is flat cells, epithelial cells, but only one cell layer thick. This is real good right here and here. So that's simple squamous epithelium. I think yeah, that's what I label it. Simple squamous epithelium. Not stratified, but the word simple. The word simple just means simple, you know, because it's only one cell thick. Epithelial tissue is either going to be stratified or it's simple. If it's stratified, it's 2 or more cell layers thick. If it's simple, it's only 1 cell layer thick. And since these is 1 cell layer thick of flat cells, we call it simple squamous epithelium. So this is a lining of the blood vessel. All blood vessels, doesn't matter if it's an arterial or a venule or a capillary, they're all lined with simple squamous epithelium, okay? There. Now the last slide, our third slide, is the mouse pinna. So what's the pinna? Look at that cute little mouse I got there. The pinna is is external ear. Only mammals have these things attached to the outside of their head, or humans got, we don't call these pinnae. Get underneath all my hair here. In humans, they call them auricles, but most other mammals have a kind of pointed looking ears. They function the same. They have to collect sound wave energy and channel it to your skull so you can hear. But you think about these structures, you know, like things appended to the skull to collect sound wave. There's no bone in there, but it has to have a skeletal element in it to give it, you know, support and structural strength. It's a different type of connective tissue to supply structural integrity. But I said not bone, something like bone, and then you can see it's cartilage. Excuse me, but a special kind of cartilage. Here it is, a section through the ear, and you can see this is the outside of the ear here, and this is the inside. Now here they say auricular cartilage. The word auricular just means of the ear. So this just means ear cartilage, okay? And here it's in the center. And then basically what from here up is the same thing as from here out, because what you're looking at is one side of the ear and the other side of the ear. So this is like the forward portion of the ear and the back facing portion of the ear, the anterior portion of the ear, posterior portion of the ear. Or on humans, it'd be like lateral part of the ear and the medial part of the ear. Okay, you got skin on both sides of your ear, right? And if you probably recognize, you see that flaky stuff? That's epidermis. You know, that's skin there, and you got it over here too. See, epidermis, this is all epidermis. We've seen these tissues already. It's gonna be stratified squamous epithelium there, then there's your dermis, and you got some fat, and then there's your skeletal element there in the middle. So here I have stratified squamous epithelium, pointing to it right there. This lining right here, you see that? That's your, we've seen that before. Now this is skin on the ear, opposed to on the tail, all right? Now in the center, that's where you have your cartilage. Now, this is not hyaline cartilage, but elastic cartilage. And elastic cartilage is different. Remember I said it's fortified inside with elastic fibers. When you think about elastic, if something has the properties of an elastic, what does it do? You know, elastic, like in your underwear or something, and it stretches out and snaps back, you know. It has elasticity, can stretch out and recoil. And so these elastic fibers is basically just forms elastics. There's a certain kind of protein that allows it to do that. And so there's only two parts of your body where you have cartilage like this, cartilage with elastic fibers. It's in the ear like that, and then your epiglottis. The epiglottis is that flap that closes over your windpipe, your voice box when you swallow. Okay, and the reason why is that it's structural, skeletal, but it's pliable. Okay, because you think about your ear, you know, you can bend your ear. It has a very distinct shape, but you can bend it and fold it up, or whatever. You let go, and it goes right back in the exact same shape. And so you want something like that. If you have these things attached to the side of your head, you know, that's pretty precarious, you know, out there. You don't want them to break off. You know, they can give, they can bend, and they can snap back. And so this cartilage has elastic fibers in it. So it's elastic cartilage. And here's a zoomed in part, and you can see very, very characteristic, because you can see the actual elastic fibers. See the little dark stringy things everywhere, very thin, delicate little bands. Those are elastic fibers. And so if you have cartilage, you should recognize this as cartilage, because that's a lacuna, and that's a cartilage cell inside of it. But around it, you got that. I think I point all of that out, or do I? I thought I did. No. But these are chondrocytes and lacunae, but chondrocytes and lacunae plus elastic fibers, that equates elastic cartilage. So there you go. There's that. Now, what else we're looking at? Okay, here I'm just kind of zoom back out to look at a different kind of tissue. This is the elastic cartilage in there, and then I'm showing you dense irregular connective tissue. Remember, we had dense irregular connective tissue in the dermis of the skin, okay? But that's not the only place you're going to have it. And there's another place that you can have dense irregular connective tissue forming something completely different. You have dense irregular connective tissue forming what's called the perichondrium. See, here's the cartilage. Perichondrium means surrounding the cartilage. And so all cartilage, doesn't matter where it is, forming your ears, forming whatever, it's always going to be encapsulated in this fibrous connective tissue membrane called perichondrium. And this is what it looks like. See this, doesn't this look just like this stuff? But it's kind of confined to the edge of the cartilage. So that's perichondrium, which is dense irregular connective tissue. And then up here is dense irregular connective tissue, but now forming the dermis. So same tissue, two different structures, okay? Look, la fin, the end. So that's it. See, that didn't even take an hour. And we went through, this is a lot faster than this lab used to be, I'm telling you, because we would, the students would get all these slides and look at it and try to figure out, one tissue versus the next. And so what I was able to do is go through it. Now listen to me. That's it. That's today's lab. And if you want to review this, you have a copy of this PowerPoint on my courses, okay? And you can go through it and quiz yourself like this. Let me fix this so it looks like this. You can go through, and this is how you study. Make sure you can identify everything like I pointed out today. And on top of that, this is a Zoom meeting, and I record all my Zoom meetings, and I'm going to upload it to your Zoom meeting folder that's on my courses. So you can view the lab. So we're all Zooming right now. Some of you might not be here live. Some of you might chose to view this Zoom meeting later because it's going to be recorded. I mean, that's fine. I don't care if you're here live or not. Because let me see this. We got 14 of y'all. That's about it. How many students am I supposed to have? So there's attending class today. I have 14 students. And I'm supposed to have, don't think I'm going to get mad at you for not being here live. I don't care. Just as long as you view this at some point, that's how you got to study. Biology 1208. Let me find you guys. I don't know how many students are supposed to be in here. Let's see. Is this it? Yeah, there you are. I already took attendance like the first day. I think everybody Zoomed the first day. 1, 2, 3, 4, 5, 6, 7, 8, 10, 12, 13, 14, 15, 16, 17. So, we're only missing a couple of y'all. That's okay. You can either view me live or those few students that are, see, 14. So I'm missing 3. 15, 16. Yeah, I'm missing 3 students. But those 3 students, hey, they can watch this recorded later on today because I'll upload this video on my courses after a while. Now, let me look at you guys for a second before I turn you loose because that's it for today for the Zoom meeting. But you think, what about lab? We got to do something in the lab. Oops, didn't want to do that. Undo that later. But anyway, or I can just ask, you can ask me questions now if you want. Do any of y'all have any questions about anything? This is your time to ask. I mean, unmute yourself and ask me if you have a question. Is there any one of you that want to go to lab this afternoon? Either at 1 o'clock. Remember, I think I said from 1 to 2 is the AI group, and then from Is that right? It's A, I forget how I did that. Half the group, and then from 2 to 3 and 3 to 4, something like that. So, is anybody want to go to lab today for anything? No. Speak now, or ever hold your pieces. So if nobody, or you can send me an email or whatever. But I will be here for a little while longer. But if I don't hear from anybody,
I do have one question.
Okay.
Just about studying. Should we study based on the images? Is it going to be a lot of visual testing, or how we should be learning this?
That's a great question. I'm glad you asked, so everybody will know. Let me get this out the way for a second. My screen is, let's see, is covered up with you guys. Let me put that back where that was. Okay, all these images I just showed you today, I'm going to use them again for your practical. Okay, and what I'm going to do for your practical exam, your midterm exam, is going to be all images of this stuff and images of next week and the week after that and week after that, etc. So it's all images, and it's all going to be naming stuff. So if you like to study, how do I study? Well, you see this right here where I got adipose connective tissue. I'm pointing that out. Make sure you recognize everything I'm pointing out. I want you to be able to recognize. So that's it. So I will see where it says dense regular connective tissue. I want you to recognize what that is. That's dense regular connective tissue forming the tendons. You should know that this is in the mouse's tail, etc. I mean, that's it. It's all identification. Okay, did that answer your question? Does everybody know? So it's all identification, all studying your slides. And the main thing that you should take home today from this lab is, okay, there's 3 slides we looked at. What are the 3 slides? And of the 3 slides, what are the tissues we saw on those 3 slides? And that's your challenge to recognize what those are. Okay.
Yeah, that definitely answered it. Just because I had a lot of on my notes information like all blood vessels are lined with simple squamous epithelium is like something you said. So I was thinking.
Yeah, that's true, and I did say that, and I kind of I will give you information like that to just drive home. If you're trying to figure out something,
right?
And well, I know that this is lined with something, and it doesn't matter where you are in the body. So we're just looking at 3 slides. So I guess for future references, but that kind of stuff, that's in my opinion, when I would teach a lecture, those are more like lecture questions. In my lab's all identification. And so, you talk about stuff in lecture and you look at stuff in lab. And so that's at least that's how I teach it. And so we're looking at stuff. And so it's really not, to me, this is like a 1-hour lab. It's not supposed to be, hard and to kill you. It's supposed to be kind of fun. You kind of look at some stuff you've probably never seen before, and it's, once you just recognize, hey, that's what it looks like. So all recognition. And so, but you have it in the PowerPoint here, you can review it like that. And then, if you have questions, you can Google these images, I mean, that's to build this PowerPoint. That's what I did. I just got all the stuff off the internet. Now I labeled everything. I just got the pictures, but you can probably find other things labeled and whatever. But I will definitely use these images for one portion of your midterm exam. And that's it. So any other questions about anything? You guys? Does anybody want to go to lab? Huh?
I have a question.
Okay.
Will we have any other like tests or quizzes other than the midterm and final?
No, you just have 2 grades in my class. You just have a midterm and a final. Your midterm is going to be, you know, it's on my courses, and it's going to be 25 questions. And let's see. And it's going to be, 25 4-point questions. So it's 100 points. And it's going to be open for like 30 minutes. So you have 30 minutes to take it. So, yeah, that's it. It will be like your midterms week, you know, whenever that, I don't know what the date is off the top of my head, but the 8th week of class is midterms week. So that it would be that Tuesday at 1 o'clock from 1 to 1:30, you take your midterm and you log on. And you'll see it's going to be a multiple choice exam, and it's going to be pictures of stuff, just like I showed you today. You know, be like this. Let me get this out of my way again. Let's pretend we're taking the midterm. I'm going to give you a mock exam here. And this is the midterm. Okay. Number 1, I'm pointing at something right here, and pretend like this is whited out. You know, that's kind of like what I'm going to do. I'm going to white this out. I say, name this structure. Is it A, the epidermis, or B, the dermis, or C, the subcutaneous layer, or what? I don't know, something like that. You know, you understand what I'm saying? So it'd be like that. And then like number 2, you know, this would be like an arrow or something pointing here. So, okay, name this tissue. Is it A, stratified squamous epithelium, B, simple squamous epithelium, you know, something like that. And the whole test will be like that, okay? And so, again, you look at the images, you study it and be able to recognize what things are pointing to and what things are. You shouldn't have any trouble, okay? And that's what we're doing this week. Next week, we're gonna do, remember, the perpetuation of life. And does anybody have that little lab manual? If you looked at this, I think it's chapter 2 or whatever. There's all kinds of stuff in there, but basically what the perpetuation of life is going to be, I think I have a PowerPoint on this, and it's probably, yeah, I think if you go to, I think all your PowerPoints are on, let's see, on my courses. Let's see, my courses. And let's see, here's my courses. Biology. That's you guys right there, isn't it? Yeah, 1208. Yeah. See, here's today. See where it says PP, it stands for PowerPoint. This is the PowerPoint for today. This is the PowerPoint for next week. Perpetuation of life PowerPoint. So I got PP for, there's 1, 2, 3, 4, 5 PowerPoints. And I think for the skeleton, there's a PowerPoint tucked away in there too. Yeah, I think that's what this is. But anyway, this is what I'm going to go over next week. And this, if you look at it, is going to be all about like cell division and like mitosis, meiosis, and embryology and so forth. And we're going to talk about all that next week. So that's a fun one to look at. But again, I'll go over it, you know, go through the PowerPoint like I did today. It'd probably take about an hour-ish. And that's it. And same kind of thing, you know, what I point out and tell you about, that's what you're responsible for knowing. Okay. So, does anybody else have any questions? How's that? Think y'all can handle all that? Okay. And nobody wants to go to lab today, right? See me face to face. If you do, just let me know. But if not, I'm gonna jet. Okay. Okay. Well, I will see you guys next week. Let me get out of here some kind of way. Here, stop share. Goodbye.